The CRISPR system is an adaptive genetic immune system for bacteria and archaea that destroys foreign DNA, preventing its incorporation into the genome. CRISPRs are clustered regularly interspaced short palindromic repeats. The loci for the system was first described in 1987, but little to nothing was known about the system until recently. As more information is gathered about the system, it has been manipulated for easier and more affordable gene editing and silencing techniques. It is important to understand the natural system to understand how these techniques work. There are three major types of CRISPR systems that all function similarly but involve different enzymes. I will focus solely on CRISPR-2, which involves the Cas9 enzyme. CRISPR involves a CRISPR array and CRISPR-associated genes, or Cas genes, which are often adjacent to the array. The CRISPR array has repeating sequences that are often palindromic or nearly palindromic, hence the system's name, and variable sequences called spacers in between. These spacers are acquired from foreign genetic material from viruses, plasmids, etc. The CRISPR array is transcribed, producing a pre-CRISPR RNA that undergoes more processing. Cas genes are also transcribed, but not shown here. Transacting CRISPR-associated RNA, or tracer RNA, is needed for CRISPR RNA processing and function. Tracer RNA forms a hairpin loop and has a region complementary to the repeating sequences of the pre-CRISPR RNA. Tracer RNA base pairs with the repeat sequences and recruits RNase 3. RNase 3 cleaves the pre-CRISPR RNA in the repeat sections, leaving 66 nucleotide CRISPR RNA molecules. The CRISPR RNA tracer RNA hybrid also recruits Cas9. The CRISPR RNA is further trimmed at the 5' end to produce 30 to 42 nucleotide mature CRISPR RNA, of which about 20 nucleotides is of spacer sequence. The CRISPR RNA and Cas9 endonuclease complex is now ready to destroy foreign DNA. The DNA that is targeted for cleavage has two main regions of importance. The first is a region called the protospacer, which matches up with the sequence in the spacer. The side that is complementary to the spacer is the target strand. The side that is identical to the spacer is the non-target strand. The second region of importance is the nucleotides adjacent to the protospacer called the protospacer adjacent motif, or PAM. The PAM is recognized by Cas9 and the CRISPR RNA spacer and the target strand bind. Once bound, Cas9 cleaves the target and non-target strand, giving a blunt end cut. The foreign DNA is eventually destroyed and viral infection is avoided. These double-stranded breaks can be repaired by non-homologous end joining or homologous recombination. This fact can be used to modify genomic sequences, and many genetic modification techniques have been developed using the CRISPR system. Now let's examine the Cas9 enzyme in a bit more detail. Cas9 is an RNA-guided endonuclease that creates double-stranded breaks in the target DNA. The structure of the Streptococcus pyogenes and actinomyces and Islendi orthologs were recently determined and published in Science in a 2014 paper by Janek et al. Shown here is the S. pyogenes ortholog. Other orthologs have similar features. The enzyme has two identical chains and each chain is bilobed. One lobe is an alpha helical lobe shown in the reds, and the other is a nuclease lobe shown in the blues. In the nuclease lobe, there are two structurally conserved nuclease domains responsible for DNA cleavage. The H and H nuclease domain in blue-gray, and the RUVC nuclease domain in blue. The H and H domain cleaves the complementary target strand, and the RUVC nuclease domain cleaves the non-target strand. A beta-beta-alpha-beta Greek key domain found in topoisomerase II, named the topohomology domain, is also in the nuclease lobe. In the alpha helical lobe, there is an arginine-rich region that is thought to mediate nucleic acid binding. RUVC, HNH, arginine-rich, and topohomology domains are conserved among orthologs, but the alpha helical lobe varies in size and orientation. Another common feature of Cas9 enzymes is a disordered region that includes the PAM recognition loops. Tryptophan residues in the loops are conserved among Cas9 proteins that recognize the same PAM. There are two main clefts on one side of the enzyme. The nuclease lobe cleft is thought to bind the displaced non-target strand because the RUVC active site is located at the bottom. The other, wider and more shallow alpha helical lobe cleft is thought to be involved in CRISPR RNA tracer RNA guide RNA binding because it is wider and has a positively charged arginine-rich region. However, the H and H nuclease domain, which cuts the target strand, is not interacting with the cleft. If the CRISPR RNA tracer RNA guide is bound to the alpha helical lobe cleft, how is it cut by the faraway H and H nuclease domain? It turns out that Cas9 enzymes are initially inactive and undergo structural changes when bound to RNA. Electron microscopy images were used to examine Cas9. Shown here are the three-dimensional EM images of apo or inactive S. pyogenes Cas9. The alpha helical lobe in gray and the RUVC nucleus domain, which is adjacent to the HNH domain, in blue are far apart. Cas9 with a CRISPR RNA tracer RNA hybrid and double-stranded DNA showed significant reorganization as seen in the second row. The larger lobe remains relatively unchanged, but the smaller lobe rotates about 100 degrees, forming a central channel. This channel is likely to contain nucleic acids, as depicted by the dashed black line in the left cartoon representation. The group decided to test Cas9 alone with CRISPR RNA tracer RNA hybrid to see if the RNA alone was enough to activate the enzyme. They saw that the central channel did form with a 50-degree rotation of the smaller lobe. This suggests that the CRISPR RNA and tracer RNA complex is enough to activate the enzyme. Not all bacteria have this CRISPR system, in part because of the energetic cost that comes with maintaining the system and the evolutionary cost of being unable to acquire certain beneficial traits, such as antibiotic resistance. The CRISPR system allows for dynamic and rapid evolutionary change in response to viral infections through incorporation of more spacers and has great potential for gene research and therapies.